this podcast, we're going to go over some of the basic structural differences between DNA and RNA, and we'll also talk about how DNA is organized in the nucleus. All right, let's start with nucleic acids. Nucleic acids are essentially polymers of nucleotide monomers. And the function of nucleic acids are to store and direct information for cellular growth and reproduction. So depending on whether you have DNA or RNA, they're going to serve a little bit different function in the body, but think of them as being like a genetic repository. It has all the information that you need for all these processes to occur in the body. So every nucleotide is going to have three parts, and we'll go into more details on these in class, but it's going to have a phosphate group, a monosaccharide, and the difference between monosaccharides tells us whether we have DNA or RNA. It's either ribose in RNA or deoxyribose, which is shown here. We're missing our OH group in the case of DNA. We're not going to really get into the structure of it, but I want you to see an individual nucleotide. We're used to seeing this double helix of DNA. If we were to unwind that and lay it flat, this is what it would look like. And so here on the backbone, we have alternating nucleotides. We go nucleotide, nucleotide. These are our monomers. So each side of this DNA is made up of repeating nucleotide units. Let's start with DNA, or deoxyribonucleic acid. And the function of DNA is to store genetic information of an organism and transmit that information. Think of it as being like a set of directions from one generation to the next. So DNA is found in the chromosomes, which are found in the nucleus. And we're going to see an image of this on the next slide, but it's small, and I want to make sure that we have a chance to talk through the basics before we get to that image. So your chromosomes are made of many different genes, and a gene is a portion of DNA that's responsible for the synthesis of proteins. So think of your DNA as being like an entire cookbook, and each of your individual recipes is something that you could bake in the kitchen, a cake, cookies. I'm only thinking of the sweet stuff right now. Each gene is similar to being a recipe. And so if I need to make a particular cake or cookie, I'm going to go to my cookbook and I'm going to flip through the pages and I'm going to find that specific recipe. Genes are just like that. If I need to make a specific protein product for the body, I'm going to go to that section of DNA that codes for that specific protein. So it's kind of like a recipe in a big cookbook. So the structure of it's a little complex on paper, but we're going to see this image soon. The DNA is wound around histones, and these form nucleosomes. So histones are kind of like a spool, and the DNA wraps around that spool like thread wraps around a spool. Your nucleosomes are twisted to form chromatin fibers, which eventually make up your entire chromosome. The number of chromosomes does differ from one species to the next. Humans have 23 pairs of chromosomes or 46 individual chromosomes. And remember that you get 23 from mom and 23 from dad. DNA molecules can contain several million nucleotides. So these are incredibly long. And DNA is double-stranded. So I remember that because RNA is single-stranded. I think DNA starts with a D and double does as well. All right, so let's look at the actual structure using a picture. So keep in mind, a strand of DNA, since it's millions of nucleotides long, is going to be incredibly long, and the nucleus of a cell is incredibly small. So we have to maximize space by packing this very large DNA into the nucleus of the cell. So we have to be strategic about it. So your DNA, your double helix, gets wound around. This is your histone, and a histone is a protein that DNA gets wound around. So that's the spool that the thread gets wound around. So these guys are going to form a nucleosome. When you have DNA and your histone together, it's a nucleosome. These are then supercoiled, and you can kind of see that coiling happening. It's coiling once, and then that coil supercoils, it twists again, and that's what forms our chromatin fiber. The chromatin fiber then is what makes up an individual chromosome. So again, the chromosome We talked about there being 23 pairs of chromosomes or 46 total chromosomes in each somatic cell of the human body. So you have 46 of those guys in your nucleus. But notice how compact and tight the DNA gets wound so that it can fit into that little tiny nucleus in every single cell of your body. All right, so RNA is a little different, same basic mechanism as DNA, but the function of RNA is to translate the genetic information that's coded in DNA into proteins needed for cellular functions. So your RNA is going to be made from that section of DNA. Whatever our recipe is or that gene in DNA, we can take RNA and we can use that little tiny section. RNA will carry the message to the ribosome and that protein product gets made. We're going to talk a lot more about this later. But the purpose of RNA is just to translate from DNA to ribosome so we can make a protein. And proteins will carry out various cellular functions. RNA is found in the cytoplasm. DNA does not leave the nucleus, but RNA can travel from the nucleus to the cytoplasm to the site of protein assembly, which is your ribosomes. RNA molecules are a lot shorter than DNA molecules. We talked about the DNA molecules being millions of nucleotides long, whereas RNA molecules are only a few thousand long. Also, they're single-stranded. So DNA had two sides to it, looked like a ladder. RNA has only a single side. And structurally, we can look over here. Ribose has that extra hydroxyl group on carbon 2. So this is RNA, but it still alternates sugar, phosphate, sugar, phosphate with our nitrogenous bases in between. So looking at this, we still have our basic monomer, our nucleotide. What's different is the bases that we use, which we're going to cover in the next podcast.